lot more aspects than just hand farting. So this is Miss Emily Jacobs, and she is actually all my inspirations in one person. So hang on, let me pull up on my notes. This is so embarrassing. So as you heard at dinner, she was the delegate from 2000 from Illinois. Coincidence, I'm from Illinois. I was born in 2000. And now she's coming back to lecture at science camp, which is amazing. Like, life goals right there. Some more fun facts about her. So I asked them like what they were afraid of. She says that she's afraid of heights, which is like slightly ironic because they both really enjoy outdoor activities, especially hiking. So whenever they're hiking and they get to a cliff, she says she really holds on to Michael's shoulder really tight. And for Dr. Michael Goard, his biggest fear is having a video not play while giving a talk. So just don't jinx it, guys. It'll be fine. And then some weird fun facts about them. Science camp was not Emily's first nerdy camp, so to say. She also went to space camp when she was 12. So, and then a little bit more bizarre, Michael once injured his head while playing chess with himself. And then a bit about them both. They're both Team iPhone. And they met each other at Berkeley doing like their neuroscience stuff. So cute. And then they also have a daughter and a dog. The dog's an Australian Shepherd named Wolfie. It's super cute, and the daughter's cute too. So give it up for Miss Emily and Mr. Mike.
Thank you so much. So I just want to begin by saying thank you to the organizers, and it is such a trip to see Josh. So I don't know where are you, Josh? Anyway, yeah, I was a delegate in 2000, and it's such an honor to be back. This is a truly life-changing experience in ways that you had no idea of. They're completely unimaginable now, but will make sense to you 18 years later. So cherish every day. So this is maybe your first split talk. So I just wanted to kind of introduce. So Michael's going to give the first half about like kind of basics of the brain and neuroscience, and then I'll take it from there. So.
All right. So thank you all for having me. Can everyone hear me okay?
Yeah.
And thank you for the introduction. That was great. / embarrassing. So, unfortunately, my biggest fear has come true. Some of the videos don't work, some of them do, so I'm going to have to jump in and out of the PowerPoint a little bit, so you'll have to excuse me. But it's okay, this will be fun. So, what I wanted to talk to you a little bit about is, so I'm going to give you kind of like a very quick crash course in what's called systems neuroscience. And so you probably have never heard that term before, but what I really want to tell you about is how our brain, which is this 3-pound kind of unassuming looking mound of kind of jellyish looking stuff in our heads, how it is actually comprised of these very delicate, kind of highly interwoven, precisely connected neural circuits. So these individual cells that have little wires sprouting off them actually form these detailed circuits that allow us to do all the amazing things that our brains do. We often take for granted all the things that our brains allow us to do, but remember that what looks like a coherent scene in front of you is actually just electromagnetic radiation coming from the sky or from these lights and bouncing off objects and going into the back of our eye. And what sounds like people's voices is actually just slight differential air pressure waves hitting our cochlea. So all these things have to be converted into information that makes sense to us in our brain. And then we have to coordinate the millions of muscle fibers attached to our bones in order to move our bodies through space and hopefully not trip over ourselves and injure our heads playing chess with ourselves. And so, this is, all of these are very difficult things to master, as anyone who's ever tried to program a computer to do any of these things or a robot to do any of these things knows. And then, of course, there's all the things that we don't even try teaching robots to do yet, which is all the stuff in between sensory perception and motor output. How do we imagine the future? How do we think about a new idea that no one's ever thought of before? How do we remember some past event from long ago? So we're going to jump a little bit into this, and so just to give you a brief overview, we tend to study neuroscience from a few different levels, kind of ascending spatial scale. So if you pick up a neuroscience textbook, oops, the pointer doesn't work, but I'll just point. Use this pointer. So if you pick up a neuroscience textbook, most of what you're going to learn about is kind of the molecular cellular basis of how neurons work. So this is how the proteins in the membrane allow ions to flow across so that neurons can fire action potentials and communicate with each other. And this is all very interesting. It's also all available in textbooks, so I'm not going to talk about it all today. Really what we're going to talk more about is this level, this kind of systems neuroscience. How do neurons kind of wire together in order to form circuits that then allow brains to accomplish all these amazing things? And really what I'm going to talk about is, how are we even looking at this? Because this is a relatively new field, and just recently, in the past 10 or 15 years, we've developed all these new, really exciting techniques that give us access to what's going on in here. And then finally, in Emily's part of the talk, she's going to jump more into this cognitive realm, which is, because all of these circuits take place within a very small space, but really the whole brain needs kind of coordinated its activity. And as Emily will discuss, even the brain coordinates activity with the rest of the body in order to actually allow all these things to happen. So I'm just going to concentrate on this right now. So why do we care about neural circuits? Why study it at this level? Why are they important? Well, as you know, if you've ever kind of looked inside your phone or a computer or any other electronic device, you have these circuit boards that have this very precise kind of wiring of all these different elements. And you can imagine if you took your soldering iron and plunked them all off and just threw them in a pile, they wouldn't do much good. You wouldn't be able to check your email on this. It wouldn't work. And so these really need to be kind of wired up in just the right way. And the brain is really no different. So we have, our neurons are cells like the other cells in our body, but they have these long structures, kind of tree-like structures called dendrites coming out, and they have another structure called an axon. And these are basically wires that connect all the neurons to each other. So the average neuron in your brain has about 1,000 other neurons it's connected to. And these, like these components, are all wired together in this very complex and precise way in order to allow all these functions to happen. So this is a slice, for example, of the hippocampus, and here the scientists use genetic tricks in order to make each single neuron a different color. It's called the rainbow technique, for obvious reasons. And so you can see these are all individual neurons, and they all have their the kind of main principal dendrite all pointing the same direction. And then over here is another layer, and they're all pointing a different direction. And these all form a circuit. They're all wired together in specific ways. It's not random. It's not a pile at all. It's much more like this. And what we need to do is kind of discover how these are wired together and then how the circuits work in order to engage various functions. So for example, this structure here, if you damage it, you lose the ability to form new memories. So very important structure right there. We want to keep it. We want to understand how it works. So what do these complicated brain circuits buy us? So we're going to kind of look at different animals in the animal kingdom. There's kind of a temptation to ascribe like an evolutionary ladder, but I just want to point out that these guys and these, they've all been around a long time. Most of them well before us, and most of them will probably outlast us. So these are very successful, little organisms. However, you can see that the brain size is increasing quite a bit. So the C. elegans has 302 neurons forming about 7,000 connections, while by the time you get up to humans, we have about 85 billion neurons forming hundreds of trillions of connections. So quite an increase in complexity there. And so what are these neurons doing? So let's start with the simplest case. Let's just look at the C. elegans roundworm. So this is a tiny little worm. It's barely visible with the naked eye. It's about 1 millimeter long and pretty much thinner. And it has these 302 neurons. So here we can be very exact. That's exactly 302 neurons. And they each have a name. We know which neurons they are and how they're connected to each other. And so this is a map of all the neurons. And we know that some of them are sensory neurons, so they kind of get sensory information from the outside world. Some of them are motor neurons, are involved in the worm being able to move around. And so the sensory is in yellow here. I'm sorry, the sensory is in blue. The motor neurons are in yellow. And there's a few neurons that aren't sensory or motor. They're what's called the interneurons, they're kind of in between. And that's shown in orange here. And there aren't very many, but there's a few, and it allows the worm to have some flexibility in its behavior. So you can give it the same odor, and it can learn to associate, very simple associations, but it can learn to associate, for example, particular odors with positive or negative stimuli. So an odor that starts out neutral can become positive or negative. So even this very simple little creature with only 302 neurons can still do some really interesting kind of complex behaviors. Now, you might say, like, okay, this worm, just it's really small, it just needs a few sensors. Maybe the reason we have so many more neurons is because we have much bigger bodies, we have more muscles to control, we have more things to sense. And to a certain extent, that is true. So as you kind of get more body weight, these are, again, a bunch of different mammals in the animal kingdom. Here, we're kind of just looking at vertebrates. This kind of breaks down a little bit if you look at invertebrates. But you see there's a pretty kind of direct relationship between body size and brain weight. Now that said, some animals actually have kind of an overrepresentation of brain size given their body weight. So for example, if you look at these animals above the line, so this means that above the line means they have more brains than you would expect given their weight. You see crows, rhesus monkeys, chimpanzees, people, dolphins, all kind of like bright creatures that we tend to think of as being pretty intelligent. It may look like it's not much of a difference, but remember this is on a logarithmic scale. So if you actually look at the ratios, so this is another way of doing it, we're basically looking at the ratio of brain versus what you would expect brain weight to be based purely on size. And you see, just some animals, cats, and notice dogs are a little higher than cats for all the cat people out there. And then, so by the time you get to chimpanzees, they have about 2.5 times as much brain weight as you expect given their size. Dolphins, it's 5 times, and then humans, it's 7.5. Now, given this isn't like an exact measure, but this is starting to reflect some of what we think about in terms of, cognitive abilities. So what do we think is going on here? And at this point, it's really speculative, but what we think is happening is that the bigger your kind of brain representation, what you're getting is not kind of more sensory neurons or more motor neurons, but all these in-between inner neurons that allow you to kind of have more complex computations, more complex kind of models of the world. So just to give you an example of this, we're going to start with a frog. And so a frog has a pretty simple brain. Even so, they have quite good vision, and they're able to, they basically have like a little visual circuit that allows them to recognize black dots on a lighter background. You can see why this would be useful for flies, flying around against the sky. And then they'll stick out their tongue and try to grab it if they see them. So, I'll show you a little video of this. So this person is playing a little ant crusher game on their iPhone. You see the frog is quite good at it. So everybody just jumps at anything that looks at all. That looks like a fly. It's almost like a computer program. If you see a dark spot, throw your tongue, bite it, that sort of thing. They don't really have a strong model of like, oh, this is like an insect, it's part of this animal kingdom, and this is the direction it's flying. The idea is that, maybe with this higher encephalization quotient, animals are able to form more complex models of the world. And, this has costs. Our brains have very high metabolic costs. Childbirth is very difficult and dangerous, and we spend a long time raising our kids before they're fully cognitively developed. But there's advantages, like we kind of know where the food is, and we know how to walk 2 blocks and then turn right, and then give the green money in your pocket to the person in order to get some chips, so we're able to carve out an evolutionary niche by having these more complex circuits. So now what we want to do is try to understand how these circuits actually work. And so the short answer is we don't know. We're getting a lot of really cool techniques for looking at them, and I think we're starting to make some progress into at least how some of the more simple behaviors might be kind of substantiated by these underlying circuits. So this is a cross-section of a mouse brain in which different types of cell markers are used. Some of them are red or purple or green. And you can see that, once again, this kind of shows some of the complexity of the brain. This isn't just kind of an homogeneous pudding, but they're really kind of wired up in specific ways. And so what I wanted to show you is some of the approaches that neuroscientists are taking to do 3 things. One, how they are mapping the brain. The first thing we need to know are where are the neurons, how are they connected to each other, which ones are projecting where, and so on. The second thing we need to be able to do is measure their activity. So neurons fire these little electrical blips called action potentials. That's how they communicate with each other. We need to be able to measure them to know what they're doing during behavior. And then finally, what we want to be able to do is actually change the activity of the neurons in a controlled way. we want to be able to say, okay, I think this neuron is involved in this behavior, so I'm going to turn that neuron off and see if that behavior goes away, or artificially turn it on and see if we can elicit that behavior. And that's really kind of the holy grail in terms of establishing both kind of causality in terms of correlation causality in terms of being able to say, okay, these neurons are underlying this function. Okay, so first, with the mapping, how do we actually map neural circuits? So this actually started about a century ago with a Spanish neuroscientist named Santiago Ramón y Cajal, and this was done in a very low-tech way. We would look in the microscope at stained sections of brain and then draw them. And luckily he was a good artist. He made these very beautiful drawings. These are actually in a museum in Madrid you can go and visit. But he was actually the first one to really describe how neurons are shaped, that they have these beautiful dendritic trees that come up and form all these connections, and made a lot of predictions that later ended up being true long before people actually have experimental techniques to measure them. But now we actually have more sophisticated techniques that allow us to really kind of see what's going on, and a lot of this is dependent on recent approaches for genetic engineering. So we can add, remove, or kind of alter genes in various genomes. You can do this with viruses. We can also do it with transgenic animals, and this gives us a lot of access to the brain. So how does this work? So I showed you earlier you can label neurons in all different colors, but let's look at a simpler case first. First, what we want to do, so I'm just going to skip by this. This is how you do it with viruses, but we're just going to go straight to transgenic mice. So one of the things you can do is take an artificial protein, or a protein that maybe exists somewhere else in nature. So in this case, they took a green fluorescent protein called GFP. They took it from a jellyfish. You know those jellyfish that float out in the ocean, they're phosphorescent. So this is a protein which appears green when it's exposed to blue light. So this is very useful because it doesn't normally exist in the nervous system. But you can take this protein and use genetic tricks to express it in particular cells in mice, or flies, or worms, or other genetically tractable organisms. So one of the first things they did was actually express it in the skin of these mice, and so they actually took a parent that was positive for this gene and another parent that was negative for the gene, crossed them, and as you can see, half the pups turned out to express this gene, half of them turned out not to express the gene. I'm guessing I don't need to point out which ones are expressing, it's quite obvious. You ever see those running around your house? Can help. But so this is in the skin. It's not very useful for neuroscientists, but it turns out we can use the same tricks to express it in the brain, in specific neurons, in specific types of neurons. So for example, this is a promoter that allows the protein to be expressed in all the neurons within a certain layer of cortex. This one is expressed very sparsely, which allows us to see the fine structure of a single neuron. We can even zoom in and see, so these are called spines, these little things on the side of the dendrite. And this is actually where the neuron makes connections with other neurons. So this is very cool, and but one problem we've had in the past is that we had to kind of take slices of the brain in order to see these neurons, because the brain is opaque. And recently, there's been some techniques developed to make the brain not opaque, and this was a very kind of crazy but clever experiment. This was done in Carl Deiser's lab at Stanford, and what they did is they applied an electric gradient to the brain and basically washed out all the fats and replaced them with a hydrogel. And by doing that, they made it optically transparent for physics reasons I won't get into. And so this is actually the same brain here and here, but here you can barely see it. You might be able to see a little bit of an outline, and you can actually read text right through it. So they're basically rendering it completely transparent. And notice you can only do this with like tissue that's been taken out and fixed and all that kind of stuff. You can't do it in like a live animal. But still, this gives you like very good access to the entire brain, so you can reconstruct it in 3D. And so this is just a quick movie. Let's see if this goes. So here they image all these different imaging planes, and so right now they're kind of traveling through the hippocampus, and then they're going to go backwards and reconstruct it all in 3D and rotate it. And what they've done is actually label different types of neurons in different colors. That's why you see the red and the blue and the green. And so this allows us to really kind of map out, in mice, we can, they're working on trying to get this to work in humans and in post-mortem tissue. But in mice, they're working on kind of being able to map like every single neuron in the brain of these animals. Just to show you another example, this really for pretty pictures. This is actually the olfactory bulb, and here they've labeled all the neurons in a whole kind of panoply of different colors. And you can really kind of zoom in and even see how the neurons are connected to each other. So a very powerful way of kind of mapping out these neural circuits. So there's a concerted research effort to kind of map out all these neural circuits, but as I mentioned earlier, just mapping them won't be enough. We also have to understand kind of what their activity is like during behavior. So it's one thing to know, like, okay, this neuron projects over to this neuron, that sort of thing. It's very useful information. But if you really want to know, like, is this neuron responsible for, the mouse being thirsty or for remembering new information, you really need to, watch those neurons while the animal is doing something. So how do we actually measure neurons during behavior? Here again, we're going to use some genetic tricks. So there's a bunch of different ways of doing this, and there's also electrical approaches where people put electrodes in the brain. I'm not going to tell you about that, but I'm just going to tell you about the approach that my favorite approach at the time. So they take this green fluorescent protein. So this comes from the jellyfish, right? And they said, okay, we want to take this green fluorescent protein, but we want it to change how fluorescent it is depending on whether or not the neuron is active or not. And so the trick they use for doing this is they know that neurons generally have a very low calcium concentration inside the cell. But when they fire action potentials, when they're active, they open these channels in the membrane and calcium ions flow in. So they reason, okay, if we can somehow measure this calcium flowing in and have it cause a change in fluorescence, then we'll have a measure of neural activity. And so what they did is they took another protein which binds calcium, and they fused these together in such a way that they basically when calcium is present, it's going to kind of tweak this protein, cause it change in its shape, and make it a little bit more fluorescent. And they did that, and they spent years doing it, and then it didn't work. It was crappy. So they tried again and again, and after like 10 or 15 years of like very careful, fine-tuned manipulating of the sequence, they eventually found one that works really well. And I was going to show that to you in a video, but that video's not working. So I'll show it to you a little bit later. So here's, and then we can, again, use genetic tricks to express this in a mouse, and we can do it in whatever neurons we want, and we can do it across the whole brain. So this is actually a mouse that I helped characterize when I was a postdoc at MIT. And so this mouse, as you can see, this is a slice through the brain. You can see that all throughout the cortex and hippocampus and thalamus, all of the neurons are expressing this green fluorescent protein. And this one isn't just a green fluorescent protein, but one that changes fluorescence depending on how active it is. So this was a really important step. So then we reasoned, okay, now we can kind of measure the activity of these cells. The problem is the brain is still really opaque. So this is kind of a top-down view of the brain, and all you can see is just like a sea of green. You can't see the individual cells, and that's because they're so closely packed that when you shine a blue light here, it not only hits the cell that you're aiming for, but it hits all the cells behind it, in front of it. And so we needed some way to be able to kind of get like a nice optical section, kind of right, one little plane, right in the brain. So here, and I won't go too much into details, but this is where we had to steal from physics. So before we were stealing from molecular biology, here we had to steal from physics and engineering. So other people, this wasn't me, but people built these very sophisticated microscopes. The one I use in my lab is this 2-photon microscope. And basically what it does is it sends, so it uses an infrared beam that's actually it's invisible, you can't even see it, and shoots it into the brain. It passes right through the brain tissue. And just where the beams cross, it excites the fluorescence. So it allows you to not have this issue where it's exciting all of the neurons all throughout the brain, but just in a very specific point. And then you sweep the beam across in order to create an image. So what does this look like in practice? So this is an example with a zebrafish. So it's actually a zebrafish larva that's embedded in agar. And this is kind of cool because they can actually image the entire organism all at once. It only has about 10,000 neurons. So this is a video. And so you see all these little dots. Each one of those is an individual neuron, and they're all blinking as the zebrafish is seeing the agar, thinking about swimming or whatever zebrafish larva I think about. And this is the same one just rotating around. So you can see it's really like a three-dimensional structure, imaging all of it. Now in mice, unfortunately, we can't image the whole brain. Their brains are just too big. There's too many neurons. But we can't image a population of several hundred to several thousand neurons at a time. And this is really cool because we can do it while the mice are actually behaving. They have no idea we're shooting invisible lasers into their brain. They're just going about their business, doing mouse things. And then what's especially cool is then we can train them to do tasks that measure certain cognitive capabilities of interest. So in my lab, we actually train them to do memory tasks. And I'm going to show you an example of this, but unfortunately, my greatest fear, these videos aren't working, so I'm going to have to put out a PowerPoint real quick. Okay, so this is what the neurons look like. So you can see they're really clearly blinking every time they become active. And we are measuring this while the mouse is playing video games. That's what in a second. So this is a mouse over here. He's using his little joystick. It works like a bike handlebar. Sorry, a lot of noise. So what they're doing is actually navigating through this maze. So we've trained them over several months to, they need to turn right when they see the blue maze and turn left when they see the yellow maze. And eventually they learn how to do this, about 80% correct. And while they're doing this, we can image activity in their cortex and see how these neurons are kind of representing this association that they've learned. And we're working on tricks for being able to both kind of help them and also impair their memory, just because we want to figure out which neurons are really essential for it. So the next part of the talk I'll tell you about is we actually are using tricks for, back to the top. So we're actually using tricks for being able to manipulate neurons in order to see whether they're actually important for various tasks. So here, so I mentioned, that we have greatly increased the ability to measure neural activity, but is this sufficient? And most people think no. In order to establish causality, we really need to be able to manipulate the activity as well. And so how are we going to do this? Once again, we're going to steal from molecular biology and from other parts of the animal kingdom in order to get useful proteins that we can put in neurons. So these are light-sensitive proteins that are involved in phototaxis. So they come from algae and from bacteria. And we take these, so they're called opsins because they're light-sensitive. And we take these opsins and we artificially express them in neurons. And then we can control their neural activity with light. So for example, this is a neuron that's expressing both of these opsins, and when we turn on blue light, it starts firing these little blips here, action potentials, so it starts firing action potentials, and we turn on the yellow light and we can silence it. So this allows us to really kind of carefully control neural activity while we're doing this. So what is this useful for? So the very first thing they did, and this was not a very sophisticated experiment, they used a virus to put this opsin in the motor cortex, and they just stimulated one side of the motor cortex. So I'll show you what this looks like. I want to point out, this doesn't hurt the mouse in any way. They're just shining light in the paint. It looks kind of creepy, but it's not painful for them in any way. So the mouse is just sitting there doing mouse things. Right now the light is off. And then you're going to see when the light turns on, they start running around in circles. And so they're actually stimulating the left side of the motor cortex, the right side of the motor cortex, which causes them to run around to the left in circles. Now, turns out they can use this, this was kind of like the dumb experiment just to make sure it's working. Turns out they can use it for much more detailed experiments. So for example, some researchers were working on how do neurons control the sensation of thirst. And they found some neurons that were particularly active when the mice were deprived of water for a few hours. So they kind of increased their activity when the mice were thirsty. And they said, okay, if we artificially stimulate these neurons, then maybe they'll make the mice really thirsty. Unfortunately, I think this is one of the videos that doesn't work. Okay, yeah, so I'm not going to show it to you. I'm just going to describe it to you. Basically what happens is the mouse is just chilling, doing its thing, and then they turn on the light, and it beelines over to the spout, and it starts licking constantly until they turn off the light. So they were able to directly control the mouse's sensation of thirst. And just to give you one final example, here, so that was by controlling neurons in the hypothalamus. These are also neurons in the hypothalamus, but they're a different type of neuron. They're actually neurons that have estrogen receptors. And you may be wondering, so this is actually a male mouse. You may be wondering, why does the male mouse have estrogen receptors? Well, it turns out testosterone is often aromatized and turned into estradiol in the brain. And so these are probably more likely acting as sensors for testosterone. And you're going to see what happens when you artificially stimulate these neurons. They put this blown-up glove in the cage with the mouse, and when they turn on the light, the mouse just starts freaking out on this glove. It's basically trying to kill the glove. And mice usually don't attack gloves, but and this is like a mouse really going crazy, jumping off the top ropes. So they basically figured out that it was making these mice highly aggressive, and it wasn't just causing the action of attack. If there's nothing in the cage, they wouldn't attack anything. And they actually trained the mice that if they went and pressed a lever, then they can get exposure to other mice. And they found when they stimulate these neurons, they run over and press a lever. So it's not like it's making them attack, it actually makes them aggressive. This is allowing us to figure out how these kind of key behaviors work, and now we're working on more complex behaviors, like how do we remember information. Neuroscientists have been able to reactivate old memories or even induce false memories for locations or places by using this type of stimulation. So in any case, just to quickly summarize, How am I doing on time? Okay, I'm just going to assume I'm doing okay. We've developed a range of techniques for mapping, measuring, and manipulating the neural circuits. And this is really leading to all kinds of breakthroughs in kind of our understanding of how different neurons control different behaviors. And eventually, the more complex behaviors, it's not going to be like one type of neuron expressing one receptor, but probably whole circuits of neurons all connected. But this is giving us some approaches for being able to get in and see how those circuits work. And finally, I just wanted to end on a note of what are we doing this for? A lot of this is kind of basic science. We just want to understand how this works. But these also have practical applications. So I actually heard that this video you've already seen before, so I'm not going to show you that one. But that actually came from understanding that dopamine neurons were important during Parkinson's, and that if they stimulate this one area, then it may be able to relieve some of the symptoms. And as you saw last time you saw the video, it's very effective in kind of immediately reducing these tremors. And I'm going to give you another example. This one always gets to me because I have a daughter who's a little older than she is. She was born with, not completely deaf, but she lost about 80% of her hearing, and they implanted her with a cochlear implant. And so again, this has to do with knowing how these neurons in the cochlea respond to sound, and basically simulating them artificially. So they take a microphone, turn the sound into neural stimulation, and use it to imitate the sound that she would be hearing.
Can you hear us? Can you hear Mom?
Yeah.
You can hear me clearly?
Yeah.
You can hear what I'm saying?
Oh gosh, it's so funny. I can hear my voice.
Have you heard your voice before?
No.
You couldn't?
No. Yeah. So I'm going to talk about these things.
So hopefully that gives you a sense for some of the kind of current really cool techniques we're using for understanding the brain, and also some of the applications that we hope to use this for. So without taking any more of Emily's time, I want to hand it over to her. At the very end, we'll take any questions you have for either one of us.
Everybody get up and stretch. Get up. Talking about the importance of tools, right, for mapping and understanding the brain. Technology is critical. I'm going to talk about something totally different, and actually a little bit of my own science at the end, because why not? So this talk is titled "The Uncharted Mind: Why Gender Diversity Drives Scientific Discovery." So, the human brain, at first glance, looks kind of unremarkable. Kind of like a 3-pound squishy walnut. But at the microscopic level, it contains multitudes, right? 85 billion neurons that are wired up in precise circuits. And these neural circuits form the basis of incredibly complex behaviors. And Michael showed you some of those. So think about it. That is capable of composing an opera. That can debate the fundamental principles of democracy. You watched the democratic debates, you saw that on display. Even more profoundly, the human brain can invent tools that allow us to see into the deepest corners of space. And maybe most profoundly of all, the human brain can invent tools to see and study itself. President Obama launched the Human Brain Initiative in 2013. At the time, he said that understanding how the brain works is one of the greatest scientific challenges of our lifetime. It's the moonshot of our generation. And it may end up being even a little bit harder than landing on the moon because there are actually more neurons in the brain than there are stars in the entire Milky Way galaxy. So around the world, there are neuroscientists who are trying to crack the neural code, like Michael explained, to understand how the brain gives rise to the mind, and to develop therapeutic solutions to devastating diseases, Alzheimer's and Parkinson's. If we have any hope of meeting those challenges, we need the best minds at the table and in the lab, inventing radically new ways of seeing, probing, and understanding the brain. And actually, that's what I want to talk about today. So this is a famous photograph. It's taken at the Solvay Conference on Photons and Electrons held in Belgium in 1927. The 29 attendees that you see here include scientific luminaries like Einstein, Schrödinger, and Heisenberg. When I first saw this photograph, I was struck by two conflicting sentiments. First, I was amazed that such an assembly of brilliant scientists whose work really shaped the world we live in could be gathered together, right, in an intimate meeting of the minds, much like this is. I was also disheartened at the people that are implicitly missing. What do you see when you look at that photograph? So of the 29 attendees, 28 were men of European origin, and only the famed Marie Curie broke precedent. And while these 29 attendees compiled an incredible list of scientific achievements, one can only imagine how much more would have been discovered had women and minorities been allowed greater access to their ranks. As the paleontologist and historian Stephen Jay Gould once said, I am somehow less interested in the weight and convolutions of Einstein's brain than in the mere certainty that people of equal talent have lived and died in cornfields and sweatshops. So today, most science departments are more diverse than they were in 1927, but not by much. There is still significant progress to make. So this graph shows the percent of female faculty in various departments at Harvard University in 2017, where give or take 4 out of 5 faculty are men. I don't need to rag on Harvard, I spent some time there, so I feel like they're a good punching bag. But actually this is true across major academic centers around the world. And in the interest of time, I'm not going to go through the long history of explicit discrimination that's kept women and minorities out of higher education. Other writers and scholars have chronicled those uncomfortable parts of America's past. Instead, I want to take a step back to examine the implicit attitudes and biases that actually impede girls' access to STEM from the very beginning. What does a scientist look like? Well, generations of children have been asked that question. In 1970, less than 1% of children who were asked to draw a scientist drew a woman. Today, about 30% do. In another study, children were shown a picture of a man and a woman, and they were asked to point to the most intelligent one. Now, 4- and 5-year-olds tend to choose their own sex, bless their hearts. But by age 6 and beyond, girls are more and more likely to choose the man. And girls who endorse a gender stereotype about brilliance are more likely to decline playing games that are described as for really smart kids. So these biases and stereotypes are affecting children's choices at heartbreakingly young ages. And over time, these are the kinds of sort of implicit biases that can impede girls' access to STEM and women's access to STEM throughout life. So, for you students in the room, some more bad news before we get to the good news. It doesn't stop at graduation. Okay, this is a study conducted in 2012 where researchers sent resumes, CVs to real science faculty around the country. This is for a lab manager position, which is often a springboard into competitive MD and PhD programs. These CVs were fake, although of course the faculty didn't know it. In fact, they were identical, except for a single word at the top of the page. The first name of the applicant was randomly assigned to be male or female. What do you think happened? Male applicants were judged to be significantly more competent and more hireable than the identical female applicants. And when pushed to come up with a starting salary, females were offered $4,000 less. Now this isn't the result of a few individuals with antiquated attitudes. That is a result of widespread implicit bias. And here's the crux of the problem. Advocating for diversity in science is not just a social justice issue. It's not just the right thing to do. Diversity in science makes science better, right? It drives innovation, it challenges the status quo, and it can change the very nature of the questions we think to ask. Here's the first example. In the biomedical sciences, like what Michael just explained to you, we rely on animal models to understand disease, to test therapeutic treatments, or to understand basic biological properties of the brain or whatever organ you're studying, right? What you may not realize is that almost all of this research is conducted in male animals. That fact was discovered and quantified by Annalise Beery when she was a postdoctoral fellow at UC Berkeley. Now she had a hunch that this was a case, but nobody had ever quantified the breadth and depth of the problem until she came along. She did an exhaustive literature review and published this groundbreaking report documenting what is now commonly referred to as the sex bias in basic science. So what she shows is that across the biomedical sciences, there's an over-reliance on male animals. This is true in neuroscience, physiology, pharmacology, cancer biology, you name it. And it's been true for half a century. It's even true for animal models of human disorders that are more prevalent in women, like Alzheimer's, anxiety, and depression. That bias has compromised the health and safety of women in ways we are just beginning to understand. Last year, the FDA pulled 18 out of the 22 drugs that it pulled from the market were done so because they had adverse effects in women. Effects that might have been uncovered had those preclinical testing included both sexes. And I argue that what's at stake here is not just the health of women, but the health of our economy, which is suffering under the enormous debt of spiraling healthcare costs. Beery's work started a movement. So by 2016, the National Institutes of Health, championed by Janine Clayton, who is the director of the Office of Research on Women's Health, they rolled out a series of policy changes and funding initiatives to ensure that future studies are balanced by sex. This was a major step forward to ensuring that men and women benefit equally from our research efforts. Right? And it's not lost on me that women played a critical role in that paradigm shift. Beery saw something that nobody else bothered to see. Right? She challenged a convention that had been passed on for decades. And in that moment, she made an indelible mark on science. So I'm going to give three more examples that hit a little closer to home. They come from my own field of cognitive neuroscience. And I would argue that cognitive neuroscience studies historically have overlooked aspects of the human condition that are relevant to at least half the world's population. In my lab in particular, I'm a neuroendocrinologist, and I study how hormones shape the brain. And so I'm going to give three examples of how menopause, the menstrual cycle, and oral hormonal contraceptives have been grossly understudied about how they affect the brain. Okay, so first, menopause. So a major driving question in neuroscience, and in particular in human neuroscience, is what happens to the brain when it ages, right, when it gets old. And the dominant paradigm in my field is to take a group of 65-year-olds and older, and to compare their brain function or brain structure to a group of young adults, usually college sophomores who need research credit. I kid you not, we're tested on a really weird sample. Now, to be fair, an incredible amount of knowledge has been generated. We've learned a lot through this approach. But that number, 65, is a historical artifact. It is rooted in the average retirement age of U.S. wage earners. It is not based on biology. There's nothing magical that happens at 65. And in fact, I would argue from a women's health perspective that it's an absurd age to choose. Because half of the world's population will go through one of the most profound neuroendocrine changes of her life a decade earlier, right? Menopause. And yet we have relatively little understanding about how the decline in ovarian function shapes the brain. So my lab, first at Harvard and now at UCSB, is really studying this midlife transition to understand how this decline in hormone production shapes the brain during this uncharted decade. And this is my graduate student treating you who really just wanted to kind of quantify the extent of this problem. So what she's mapping here in blue are all the studies published with, so I predominantly use magnetic resonance imaging, MRI, or fMRI if you want to look at brain function. And so fMRI really kind of landed, was adopted early in 1995 and is used extensively now. And so what you see in blue are the number of aging studies that use that kind of classical paradigm of 65 in young adults. And there's thousands of studies that have been published over the last 25 years. If you squint, you'll see in orange the number of studies that have addressed this midlife window, right? This reproductive aging as opposed to chronological aging. So there's a lot of room for improvement. And I would say that, this is particularly troubling given that by some estimates, women are at twice the risk for Alzheimer's. And we don't know why. And maybe we should start studying women if we want to figure it out. Okay, so I'll give another example, which is, this is another graduate student of mine, Laura Pritchett, and she had this very Marie Curie moment where she wanted to do some self-experimentation. I hope her end is not as severe as Marie Curie. So, again, I study hormones in my lab because the brain is an endocrine organ. So half of the cells in your prefrontal cortex contain estrogen receptors. Half. So this suggests it's a major organizing principle of how the brain works. And yet, we don't study hormones in neuroscience. So neuroscientists are basically neck-up people. We are obsessed with how the brain works, but we get so kind of enamored with the complexities and the unique capabilities of the brain that we forget it's part of this larger biological system, right? The body. And hormones are a perfect example of this sort of brain-body interaction. So hormones are released through endocrine glands throughout your body, they travel through the bloodstream, they pass through the blood-brain barrier, and they can affect brain structure and function. And we know this from animal studies, but nobody in my field has bothered to understand these relationships, and I think that that's a mistake. And so what Laura did in the first of its kind is this dense sampling, deep phenotyping project, where we actually scanned her brain and drew her blood and did a bunch of other stuff every single day of a complete menstrual cycle, right? A complete reproductive cycle. So here she is getting her blood drawn and going into the scanner. And here's why. Because over a kind of canonical 28-day cycle, estradiol, which is the major form of estrogen in mammals, including humans, shows about a 12-fold increase. This is right around the period of ovulation, and it goes back down, so there's these sort of sinusoidal changes. Progesterone levels show an 800-fold increase. So in the last 2 weeks of your cycle, progesterone levels skyrocket. And these are neuroactive hormones, right? They bind to receptors in the brain, they change aspects of dendrites, which you just learned about, so microstructure. And we know in rats doing overactivization in hormone back studies that they affect behavior. So why are we studying these in humans? Well, Laura did. And one of the first things she showed is that this change in estradiol over this 28-day period so increases in estradiol over time are associated with greater functional connectivity across the whole brain. So what what's represented here in yellow are just sort of the magnitude of that change. So this is incredible because it's the whole brain that's being affected. And here's the problem, right? Women make up 50% of the study population, but less than 1/10 of 1% of human neuroimaging papers published in the top journals in the last full year considered endocrine factors in their design. So I would argue that we are missing essential features that shape the brain. We are not going to understand how the brain works unless we take these into account. Now, I'm giving examples from women. Obviously, men have hormones too, and we're actually interested in that, but because I get to decide if those movies talk about women. Okay, what about, so that was brain function. What about brain structure? So in an analysis led by my postdoctoral fellow, Caitlin Taylor, she actually shows that change in progesterone over the cycle leads to observable changes in brain morphology or brain structure. And we looked at this by using a really high resolution scan of just the hippocampus, because the hippocampus outside of the hypothalamus is probably the densest region of the brain. It has the densest number of hormone receptors. And so here we're looking at sort of sub-millimeter resolution of the hippocampus because it's not all one thing. It contains these subfields. If you've taken a neuroscience lecture class, maybe you know that it's kind of multifaceted. It's got all these subfields. And so we can look at that through a high-resolution scan. And what she found is that those changes in progesterone levels actually show an observable change in regions of the brain. So shown here in yellow is the entorhinal cortex. We also observe it in some of these other subfields. What's so fascinating to me is that this region of the brain, entorhinal cortex, is the first part of the human brain to show neuropathology with Alzheimer's disease. And here we show that it's under the regulation of hormones. So this is, we are just scratching the surface because we're really going it alone here. And we need a lot more people to understand what that means and how it can inform treatments. But the point is, these are the kinds of questions we need to be asking. Okay, and I'll end with a final example that maybe hits close to home for you, which is, giving examples of how endogenous hormones shape the brain. Well, what about exogenous hormones, meaning the kind that we might take or consume either unknowingly, like through endocrine disrupting chemicals in our water, or knowingly, right, like the birth control pill? I want to preface this part of the talk by saying, hopefully I'm not being too political here, I think the birth control might be one of the most important human inventions ever made, because it gave women control over their own reproductive function for the first time in human history, which led to economic independence and financial gain and gains in the workforce. I'm a huge fan of the pill, okay? Just to lay that groundwork. But I would also argue that we need more and better research to understand what it does to the brain, because it turns out that that is not a requirement by the FDA to check safety profiles of the drug. The pill, so oral hormonal contraceptives, or commonly known as the pill, is used by 100 million women worldwide, 10 million women here in the United States. And yet we have relatively little understanding what it does to the brain, even though my whole lab studies the effects of endocrine function on brain morphology and brain function. So that's alarming. So my postdoc decided to sort of, when she came into my lab, convince me that we had to study this. And at first, I will say, I was a little resistant, because I did not want to be a Breitbart News headline and say the pill changed the brain, because you can imagine how people who are not supportive of Planned Parenthood would run with that. But I realized that ignorance is not bliss, and we can recognize that the pill is important and also try to understand what it does so we can innovate in terms of the kinds of pills that we're using. And so what she has developed at UCSB is a research consortium that basically pools scans. So we're trying to develop sort of a big data approach, because you can imagine trying to ask this question, what is the pill of your brain? There's lots of confounding factors, right? We have to consider age of initiation. Do you start when you're 13 or when you're 30? We know the brain undergoes protracted development, so maybe starting at younger matters more. What's the duration of use? If you're on it 6 months versus 10 years, there's probably a difference. And then formulation, right? So if we get the brand name, we can reverse engineer what the formulation is. And so, long story short, we've been building this database. And what she's found in about the first 100, 150 participants is that there are demonstrable differences in brain, just at the gross level brain morphology. So here what she's looking at are current users of the pill compared to And of course, there's this middle group of people that have used it at some point in the past that aren't currently using it, and I'm not sure what's doing now. And so here she's showing differences in cerebellar volume, and the duration of use. The longer when they've been on the pill, the larger the changes that we see. Again, this is just scratching the surface, really sort of simple comparisons. But we actually use this as pilot data to build the University of California Women's Brain Initiative, which is the first large-scale brain imaging consortium that is dedicated solely to women's health research. There are other consortiums out there that you might have heard about, so the Connectome Project, the Harvard Gene Superstructure Project. And we didn't want to reinvent the wheel, right? So we went to them first, we developed collaborations, we got their data, because they said, sure, we asked about the pill. But it turns out the questions that they asked were so short-sighted, because it wasn't designed by a woman. So they asked, I think it was, have you ever been on the pill, yes or no? And then the follow-up question was like, if so, why? I don't know why. I don't know when they started, what the brand name was, you can get formulation. So anyway, we decided that if nobody had done it, we're going to do it ourselves. And so what we've done now, we've now reached out to Berkeley, and we have data collection sites there. And this is part of our larger goal to get sites at all of the University of California campuses that have a research-dedicated brain imaging center. So we're basically pooling these brain scans so we can get 10,000 individuals over time to do real population neuroscience, ask questions at the intersection of the brain and behavior as it's relevant to women. So, here's I guess, the crux of the problem, is that scientists cannot answer questions we don't see. And for a field that has traditionally been led by men, it's likely that menopause, menstrual cycle, birth control, these were never visible problems or issues, right? I say problems not like bad, but just great factors. Just like it didn't seem obvious to only study male animals. Until somebody comes along and questions those ingrained assumptions. So what are all the other questions we can't see because we lack the experience to even imagine them? Science has to represent society, especially since the bulk of our academic funding comes from you, right? From taxpayer dollars. So we need a pool of scientists who bring radically new perspectives to the table. And if history is any evidence, when women gain a voice, and when we step into positions of power, it transforms science. So I can only hope that when future generations look back on a photograph of the most influential neuroscientists from the present day, they see a group of faces that more faithfully represent our social makeup as a whole. And if they do, I think our moonshot will be in reach.
How close are we to mapping the human brain as a whole?
Not close. So they're working on, so it depends at what level of resolution. So the kind of gold standard is what's called a connectome. So it's, you know where all the neurons are and how they're connected to each other. So this has been completely accomplished for two organisms. So one of them is that roundworm I showed you that has 302 neurons, and the other one is a larval sea squirt that has even fewer neurons. So we'd like to get into some more complex organisms. One problem is that when you get to something like a mouse or even like a fruit fly, the neurons aren't the same between each organism. So it becomes kind of like the Human Genome Project, where you're basically looking for like probabilities of different neurons being in different places and probabilities of being connected. And so for the mouse connectome, they've made a lot of progress and they're getting closer and closer. For humans, we're still quite far off, but we're kind of mapping, so people in Emily's field are kind of mapping the course structure and connectivity, like which brain areas tend to talk to each other across people. But we're talking, it's not an impossible problem, but it's probably some time away before we have kind of a complete map of the human brain.
So when we were talking earlier about using oral contraceptives, so when you were saying how using oral contraceptives changes brain morphology, what are the effects of that on everyday behavior and thinking?
Yeah, that's the biggie, right? So how does it affect function? Because who cares about how my brain looks, how many wrinkles are there, but like, is it going to affect function? And there have been small scale studies, this is the problem, right? So it's like 20 people in one group, 20 people in another, to suggest that there are changes. But I would say that it's pretty crap science. And so the short answer, this might be a recurring thing, is we don't know. And so you should look.
Do you think that fixing the gender disparity is going to come from a policy perspective, or is it going to be a cultural shift and social shift? And for either, what do you think is the best thing we can do as individuals to try to fix the problem?
Be nice to each other. Form science clubs that are inclusive. When you're in that intro physics class, don't dominate discussion, even if the woman is a little, more hesitant to answer the question. Just recognize that your future is dependent on diversity in science. And I hope that's the biggest thing. Is again, it's not just like a nice thing to do. It's actually critical for science discovery. And so I think if we're all aware of that, and then the issues with the biases. When you become PI of your lab, recognize that those biases exist, and then check yourself. When you have resumes floating across your desk, and you have to decide, I think just being aware of the fact that there is a bias is huge. That's the first step. You can just shout it out.
Okay, so you study the effects of hormones on the brain, correct? In terms of pregnancy, do you look into that? Because when you get pregnant, obviously your hormones do wild things and increase in level, but does that affect, so kind of like the theme of younger generations having kids later in life and maybe having less kids, do you know if that affects just the overall brain?
Yeah, so two things. So one, that was 2018. We are, fingers crossed, about to run what we're jokingly referring to as 2018 baby. So we're actually going to image a woman about every week of a 40-week pregnancy to understand how the brain changes. Now we have some evidence. It was just a splashy paper in Nature, I think last year, where they studied a group of women right before they got pregnant and then immediately after delivery and then a year later. And they show really sweeping, because you're right, that this order of magnitude levels of change that you're seeing in these hormones. And there were clear changes in gray matter. So actually, gray matter volume in the sort of circuit of the brain that's called theory of mind shrunk with pregnancy. Now people always think, oh my God, your brain is getting smaller, that must be a bad thing. That's not always the case, and I'll give you an example. So in adolescence, during puberty, right, another period of major hormonal change for boys and girls, you also see this fine-tuning of synapses. So white matter might increase, gray matter actually gets turned away. And you can think of it, and that's a good thing, right, because it sort of gets rid of extraneous synapses that we don't need and strengthens the existing connections that are there. And so you can think of it like Michelangelo's David. You start off with this hunk of granite or marble, and you chip away to reveal the underlying beauty. And so there's periods in the human life, and we're just beginning to understand this, where these hormones are actually really critical for sculpting the brain in these really beautiful ways. So we've known for some time about adolescence, and just 2 years ago, this paper got published showing that the same things happen during pregnancy. So that theory of mind circuit gets really kind of like honed, and it's actually, to go back to sort of a previous question, predictive of function. So in mothers that showed the greatest degree of change in that region, they actually had greater sort of empathy using these kind of social like tests with their babies, suggesting that this is really important formation in which the brain changes. And probably what we're going to be seeing is that menopause is another period where the brain kind of reorganizes in interesting ways.
How did you find this intersection of social justice through science as a postdoc?
Yeah, so I like to joke in my lab that research is research, that we ask the questions that we see and that are obvious to us. And, I've always been a feminist, and I think when Annalise, and she's a friend of mine, when we were both, what were we, grad students at Berkeley at the time in that paper, when she was working on that data, and I was appalled by the fact that the questions that as a field we were asking were not reflective of who I was, and I'm just one slice of the pie, but what about everybody else? And so it's just, I think, being observant and recognizing not what interesting question you want to follow, but like what question isn't there, and carving it out to make it grow. So just, back to awareness.
I have a question for Michael. So, let's say theoretically we have a human brain that's higher than that. Do you think that the morphology alone of that brain will nominate its size into personality, intelligence, and cultural background?
Probably not. So, can you repeat the question? So the question was, let's say we had an entire map of the human brain. Would that map give us insight into the personality, cultural background, etc. of that person? And certainly at this point the answer is no. And that's, I think to understand how a brain would behave, you'd have to have kind of like a complete model of, how it would act if you just started running. Now that information probably is in there, in the connections between all the neurons in the brain. How do we know this? Well, if you turn the brain off, if you go under anesthesia or go into a coma and then turn back on, you're still the same person afterwards, right? You might have a little blank space while you're out. But those hard connections between your neurons are really what make your memories and your personality and all that stuff. But in terms of us being able to read it out, I think we're a long way from that.
Okay, so I want to go into philosophy right here. And so essentially, there are people who think that emergence is a thing and then there's a reductionist. So, like, in terms of the brain is what consciousness is talking about. So, do you think, like, brain structures of the neuron are, like, themselves, they're basically that's like it? Or is it, like, more, if you just sum the neurons, it's like one of the self-response, or just
So the question was, is consciousness just due to neural activity, or is it kind of an emergent property that somehow is beyond just neural activity alone, or just arises from coordinated neural activity? I'll take this one. I don't think we really know. I mean, everyone has their theories online, but I wouldn't say there's any scientific backing.
This is answering your question, really. But I will say that I teach hormones in the brain class, and I joke that by the end, nobody believes in free will anymore, because the more we learn about, so has anybody heard of Odin's curse? I don't know, Odin's curse. So it's like one of the all-time creepiest diseases. And basically, it's a lesion in your hypothalamus that stops you from you lose the capability of doing automatic breathing. So what do you think you die from if you have Odin's curse? It's not asphyxiation. It's actually sleep deprivation. Because every time you try to fall asleep, you choke yourself back open. And there's examples of like people getting lesions to their amygdala and changing their personalities overnight. So the more we learn about it, the more we realize that aspects of our human behavior are governed by the brain, and probably everything that we don't know that we classify as sort of mystical or just things that we haven't figured out yet. But I think probably we will one day.
So you talked a little bit about emerging research and how some of the traumas and time into your post-menopause and further or get further into Alzheimer's. Have you started to
And depression is a major comorbidity of Alzheimer's disease.
Can you repeat the question?
Oh, yeah, so basically hormones and depression. So what do we know about hormones' role in other kind of disorders? And we know that secondary hormones, and in particular estradiol, play a major role in depression and sort of sculpting the neural circuits, stress circuitry in the brain. So yeah, there's a pretty cool research around that. Depression is about twice as prevalent in women, so there's a sex difference in prevalence and incidence rates there. And interesting, we can learn out about it. Okay, I don't know how much we can just keep going. Okay.
So I have two questions. One is, can you, I know you can stimulate certain neurons, but can you sting specific neurons? And the second one is, when you do get that 3D model, is it a computer program that identifies the neurons, or is it a person saying this neuron is here and now?
Right, so the first question was, can you label specific neurons? Is that?
State them.
State them. Yeah, so there's the same techniques we use for clearing the brain. You can actually use it for putting in antibodies that have different fluorescent labels on. So yeah, we can figure out what type of neurons they are, what type of neurotransmitters they release, a bunch of other information about them. And that technology is kind of growing all the time, now you can make really nice 3D models with all that information tagged. It's mostly being done by computer now because there's just so many neurons. You know, even a mouse brain, we're talking, million, hundreds of, roughly 100 million neurons in a mouse brain. So that would be a lot of people sitting there at the computer clicking. So it's mostly being automated by computers. And there's, some errors, but it's low enough. It's probably about equal to what the human error rate would be.
So the amount of interneurons increases as body mass increases. So does the amount of myelin sheathing also increase to speed up those connections?
Yeah, so the amount of interneurons increases, it actually doesn't necessarily increase with the amount of body mass. It only does for those creatures that are kind of above the line. So like the chimpanzees and humans and crows, and so they tend to have more of the interneurons relative to what you'd expect for body mass. Now in terms of myelin, that tends to correlate more with the size of the animal, and individual neurons even within an animal have like huge differences in the amount of myelin. So for example, a lot of neuroscience was done on this one squid neuron called the giant squid axon, because it's so thick you can actually see it by eye. I mean, usually these are microscopic, you only can see them with a high-powered microscope, and it's because it's involved in this escape response. So it needs to travel, the action potential needs to travel this really long distance and drive this muscle really strongly. So you have this huge axon that does it, but then other neurons in the squid are tiny. So there's tons of variation.
10 minutes for questions. 10 minutes for questions.
10 minutes.
I have two questions. First, I think you mentioned that male mice are less expensive than female mice. Can you explain why that is? And second, humans are the only animal that have periods, so how can we do without when twice we don't have the same?
So did everybody hear that? So one is that I said that male mice were cheaper, sorry, to house, and then humans are the only ones that have periods, so how do we do with that? So the first one, so that, I'm super glad you brought that up. So, a lot of times I get like, why was there this convention to only study males for half a century? And there's usually three reasons held up. One is convention, so what my professor did, and so those are the lab techniques I learned, and I adopted them, and et cetera. Well, convention's an easy one to do away with, so let's not worry about that. The second one is cost, that if we're going to increase our sample size and include females, we need more cages, and we have, so money, right? Money's limited, we have to go to the NIH for money, so that's a tough one. And then the third one is this issue of variability, right? So there's this knee-jerk assumption that female animals were inherently more variable because we have this pesky menstrual cycle. So the first one, cost, it's not really more to house a female than a male. I mean, it costs to house animals, period. So that one is kind of a misnomer. The third one, this idea of variability, turns out, here, all you good soon-to-be scientists pay attention, nobody had ever empirically tested that claim, ever. So what Annelise's crew did next to combat that is to do it themselves. So they took something like 600 different endpoints. So in mice, they looked at studies that had looked at cell signaling properties all the way up to behavior of an animal. right? So like all those levels of scale that Michael talked about. And they weren't interested in like the mean values between the sexes, like are men up here and women down here, vice versa. They didn't care about mean values, they cared about the variance around the mean, right? The coefficient of variation. And what they mapped is just basically whether females were more variable or males across biology, right? Across the animal organism. And so there, I don't know, there was like 400 or 500 different variables that they looked at. And do you know how many of them females were more variable than males? 0. Here's the kicker. You know how many which males were more variable than females? Like 2 dozen. So by that, we would have totally flipped the convention on their head and not studied males, right? Had anybody actually bothered to look. But here is where these gender ideas come into play. And we think a menstrual cycle, no. Obviously, I've just shown you data that cyclic changes in hormones can cause or create cyclic changes in the brain. But that's one source of variability in a sea of variables. And it turns out there's probably more variables that are affecting males than females, right? So we have to think about what are the underlying assumptions that are driving these major research practices that have undermined women's health, as I said, for half a century. So if you come across something when you're like reading the method section of the paper and you think, why are they doing that? Like, go look and ask why. Because it may, you may change the course of, biological science. Always.
I was just wondering, it was a less technical question, maybe a little broad, but if you could speak to any instances of like bias in the workplace of your education or like discrediting of your research, just that you've experienced and how you handled it.
I don't know. I mean, I went to Smith, not per se because it was a women's college, but because it, at the time, 18 years ago, it had a neuroscience program and it was super kick-ass. And so, but I realized that in retrospect, being a woman in the hard sciences at a women's college was incredible because I didn't deal with any of that, like, being the only female in the intro physics or in my quantum mechanics class. No, like, everybody was a woman, and so it didn't seem weird, it just felt normal. And so, and I went to Harvard a little bit in college too for my junior year, and that is when it all crystallized for me, because my best friend who started there and finished there was also a neuroscience major, and she had a completely different experience. Now, these are ends of one, but still, I saw her personal story as being very different from that, dominated field. So I would say I cruised through kind of unaffected because of that. I don't know. I mean, I can, when you're on the job market, and I won't say where, but, we were blessed to have multiple offers at multiple institutions, and there was one that offered me, so, you get like a startup package and lab space and all this stuff, and like a salary. And in me, pretty identical, I was like, I don't use it in my TV, like, I don't know, maybe one's better, but it's clearly Harvard. Sorry. But I actually had somebody come up with an offer that was $2,000 less salary for me. And it's just like an annoying amount. It's not like 10, it's not like it matters. It was just like, what? And I actually went up to the chair and I was like, is this a typo? I'm just gonna assume it was a typo because that's super annoying. So yeah, I guess there were certain instances like that, but you kind of just joke about it and move on, and you get allies, and marry yourself a feminist husband.
Do you have any theories on why males are so much more variable than women?
So the current theory, and people don't really know, but the current theory is women have 2 X chromosomes, males have X and a Y, and the Y chromosome is actually really, really short. It's a very small chromosome, and it just has a couple genes that are basically responsible for kind of generating maleness. So as a result, the X chromosome for women is kind of duplicated, and there's this phenomenon called X inactivation, where genes will actually inactivate like the other side of the X chromosome. And as a result, the thinking is that kind of reduces variability. So different cells will express different copies of the X and inactivate the other one. And so you're kind of averaging the two X chromosomes together. While in males, they just have the one X chromosome. So as a result, you're going to get kind of higher variance. That's the thinking. It's a hard thing to prove it, but that's kind of the current thing.
3 more questions?
Let's see if I can use the microphone. I generally think like artificial neural networks whenever I think of natural neural networks, and there's always like a weight in terms of connection between the two nodes. Is there any way in your neuronal mapping can you account for an exposure versus an inhibitory synaptic connection between two cells in the mapping tool?
So it's really difficult. So people are trying. There are small anatomical differences between excitatory and inhibitory synapses, and small anatomical differences between strong versus weak. So that's the other thing. How do you tell like a barely there synapse? So we know their synapses are actually silent, so they form a connection, but they're not actually active in normal circumstances. Versus like very strong. Some synapses which are amazingly powerful. I'd say all of it's pretty rough at this point. So there's some other approaches people are doing for basically doing functional mapping. So that's where you kind of measure the connections between all the neurons first, and then you barcode them in some way, and then later you can kind of figure out how they're positioned, how they're connected. But it's kind of in its infancy at this time, but that would be very important information to have.
I was wondering if it's been looked at, if it's been looked at for the potential of unconscious bias, not only against women but also minorities in science, and possibly addressing that not only as, like you said, for Toby's question, where it's more of just thinking about it and being aware of it, but possibly how individuals are raised or immersed in an environment.
Yes, there are lots of people thinking about this in the social sciences and social psychology. We're social creatures. We live in a social world, and I don't know if this is an investor, but we think about this a lot. I'm raising a daughter, right, who just turned 6, and I'm like, I'm gonna do the intelligence test. Weird. But you start to catch yourself, right? Do you talk about policemen or do you talk about police officers, right? It's embedded in our language, in our social media, in our TV, and so I think again, you can start to piece together these causal chains and of how these biases can become so insidious, and how do you counteract them? And again, awareness is a little bit of a silly thing, but actually I do think about it. Awareness and an action on that, right? So investing in, there are people that do this, who do there's environmental impact assessments, EIAs, they can do the same for media, right? And there's whole groups of activists that lobby Hollywood and screenwriters to display diverse casts or tell diverse storylines, or Glee did this really famously and sort of change the dialogue around trans. So I think it's going to take that and bottom up and top down approaches. And again, I think people like Janine, right, who are in positions of power so she's the head of the National Institute of Everything goes with NIH, right? It is the major funding body for most biomedical sciences, that and NSF here in the United States. I don't know what our budget was last year. It's $1 trillion. I don't know, it's a lot. So Francis Collins is that current director of the NIH, and Janine, who I showed, convinced Francis that he had to change the policy. So now when you apply for grants at NIH, you have to have an inclusion policy. And if you're doing preclinical animal research and you say you're only studying one sex, the onus is now on you to justify that. And if you don't do a good job, you don't get the money. If you don't get the money, you're not going to stay in science, right? So, and even journal editors or reviewers, so if a study was only published in males, somebody had this sort of tongue-in-cheek thing is that you should sort of tack onto the words to the end of your title and say in males, or just lots of ways being discussed to change the sort of existing biases, but also to be aware of the other ones that matter.
Thank you.
Last one.
If a human brain were to be constructed somehow based on a completed connectome of the human brain, would that constructed human brain be considered an individual? Like, what are your thoughts on the ethics of that?
That's a good one. I mean, I think at some point, if artificial intelligence gets sufficiently complex, then at some point, you would get artificial consciousness, and at that point, I think you have to kind of treat it like biological consciousness. I don't see that it matters if it's made out of proteins or silicon in terms of how you would kind of treat it as an organism. But probably people would disagree. Thank you all for all the great questions.